atherosclerosis and lipid metabolism for a very long time. And today, actually, there's a very cool connection between, you know, the non-human interaction and the human interaction and microRNAs and inflammation, which I think we're all really excited about. And Casey and I have known each other a very long time, and I like to say that we're in the same like cohort. We grew up, came up together, so we're so it's a lot of fun. So take it away, Casey.
Okay, thank you. Share my screen. Okay, are we good?
Yeah,
that's
good.
Okay, so today I'm going to tell our story on LDL and its transport of microbial small non-coding RNAs, and how we think this is a driver of inflammation within the lesion. And we'll talk a little bit about some of the cellular processes underpinning this. I just wanted to start with the overall model, so we're kind of on the same page as we go through some of the details. We haven't worked out all the steps here, but we think that lipoproteins can acquire extracellular RNAs, mainly from the mucosal interface between our environment, microbiome, and the immune cells within those tissues. So we think that as bacteria and other pathogens are broken down, it releases a lot of extracellular RNAs that lipoproteins can sweep by and pick up. They will remove those and excrete those from the body. We don't know a lot about that, but what we do know is that when LDL starts to accumulate in places where it's not supposed to be, like the lining of the artery wall, it can elicit immune reaction. And we think that these foreign RNAs, these microbial RNAs, will drive some of the inflammation in the arterial wall. And then HDL, I'm not going to talk about HDL today, but we think that HDL will remove some of this burden of extracellular RNA and bring it back to the liver and potentially the kidney for excretion. So that's the general model that I'm going to talk about today. So if we were to start with our cartoon of Afro, you know, for years we've kind of used this deposition slide where LDL will deposit cholesterol, bioactive lipids in the arterial wall, and HDL will come by and remove those. So it's just a very similar story. The parallel here is that we think that LDL can deposit microbial products in the arterial wall, and that HDL can remove them. So it's a very similar to the cholesterol story, but just in a parallel track. So this research is mainly driven by Ron Allen in the lab. Ron started with me as a postdoc, and he's now an instructor. Ron's doing great. He's actually on the job market now, so he's looking for his first faculty position. His work in the lab was on the role of microbial small RNAs, mainly on ApoB-containing lipoproteins, and how this factors in atherosclerosis and cardiometabolism. Okay, so over the last about 5 to 7 years, we really kind of turned our attention back to native LDL, more so than oxidized LDL. So we're really interested in the role in cargo of just native unmodified LDL. And you know, back in 2009, this really caught my eye in that majority, or at least half of the patients that come in, they're admitted to the hospital, have normal LDL cholesterol levels. And so we clearly know that LDL is a causal factor for atherosclerosis. And LDL cholesterol is a primary therapeutic target to reduce cardiovascular events. But there's still something special about LDL itself, the particles themselves. Of course, we were very excited about the CANTOS trial and how it demonstrated that you can reduce cardiovascular events in absence of lowering plasma cholesterol. But we're also interested in the residual inflammation within this therapy. But of course, the CANTOS trial proved the inflammatory hypothesis of CVD after years of postulation. But the downside to inhibiting cytokines or going after cytokines was observed with a statistically significant increase in fatal infections. So instead of going after the cytokines, which we think are kind of downstream of the inflammation, we wanted to start to address some of the upstream stimuli and kind of look and see if there's anything on LDL that would confer this early stimulation of the inflammation. So we took native LDL and we treated PMA stimulated THP1 macrophages. And we did, this is at 500 micrograms of LDL. And after 24 hours, we did mRNA sequencing. And oh gosh, LDL drives many changes in genes in macrophages. And we're able to list a lot of the kind of key genes that we found. A lot of those are familiar with people, the cytokines and things like that. But if you do a transcription factor analysis, the transcription factors that are most active in this data set are the ones that you would expect: NF-kappa B, C-Jun, and some other transcription factors. So this really, for us, solidified that or justified us looking into the role of native LDL instead of oxidized LDL, going back to the native LDL activation. So the LDL that we use is non-oxidized. We kind of test the LDL here with agarose gels, but also with the T-bar assays, just to make sure that we're dealing with minimally modified, or sorry, dealing with native LDL. So we did a concentration, dose-dependent experiment where we took THP1 cells and hit them with native LDL, starting with 200 micrograms up to 1 milligram. And after 24 hours, there was a dose-dependent increase in the mRNA expression, a lot of the key cytokine genes, IL-1 beta, IL-6, TNF-alpha. And so then we looked at the temporal dynamics of this, and native LDL was found to increase pro-inflammatory gene expression as early as 3 hours for some of these genes. There is a little bit of an increase and then waning of the gene expression by 24 hours, but a lot of our data is at the 24-hour time point. So based down low, you can see that FLIN2 is actually going up, as indicating that the cells are receiving the LDL. One of the interesting things that we're observing is that IL-10 is actually suppressed with the LDL. But we're focused mainly on the cytokines at the mRNA level here. So this was at 500 micrograms of LDL. At the protein level, we were doing some multiplex Luminex assays, and we would sample the media about 24 hours after the LDL treatment, and a lot of the cytokines are starting to accumulate there after 24 hours. TNF-alpha, IL-6 are readily detectable in the media, THP1 cells. So this is very basic experimentation here. So the hypothesis based on this, kind of what we know about the LDL RNA, is that we hypothesize that exogenous microbial RNA on the LDL promote inflammation in the macrophages. And so the mechanism that we went after is Telopreceptor 8, which is an endosomal receptor that I'll circle back to in the second half of the talk. But this is the mechanism that we think is happening. So maybe there's a little bit of LDL receptor mediated uptake, but we think the main route is through this non-saturable uptake of pinocytosis. LDL can accumulate in the arterial wall to pretty high levels, around 2 mg per mil in the arterial wall. And at that level, we think that the macrophages are just sampling their environment and bringing in quite a bit of LDL into the endolysosomal system. There, as LDL is broken down, the microbial RNA on the LDL is exposed to these single-stranded RNA sensors, Telopreceptors 7 and 8. But in the macrophage, we think 8 plays a pretty strong role. So let's talk a little bit about the RNA on LDL. So back in about 10 years ago, we found that LDL transports microRNAs. And over the last decade, we've used small RNA sequencing to really characterize and profile the RNAs on LDL. And a few years ago, we released a study. We published a study showing that LDL, ApoB-containing lipoproteins, are highly enriched in microbial RNAs. So we think that the microbial RNA on LDL really fits this kind of non-standard risk factor for stimulating atherosclerosis. This is a recent review by Peter Libby's group, and looking at some of these new mechanisms that are new factors that influence inflammation in the arterial wall. We have a new reagent in the lab that we're excited about called RNA Select. And Mark Castleberry joined our lab about a year ago from Sean Davidson's lab. He's a postdoc in the lab, and he's been getting the SATO assay working. And we like this assay because it allows us to quantify the total amount of RNA on lipoproteins without having to isolate the RNA. This is basically a cell permeating RNA dye that we can use with a standard curve. So when we started looking at the different lipoprotein classes, we'd isolate lipoproteins with density gradient ultracentrifugation, and then quantify the amount of RNA, normalize it per protein. That it seems that LDL has a little bit more RNA than VLDL and a little bit more RNA than the HDL. And if you look at the human plasma across size exclusion chromatography fractions, it suggests that the ApoB lipoproteins do indeed have quite a bit of the extracellular RNA in plasma. So HDL, the way we have this FPLC set up, the HDL range is stretched a little bit, but there's also RNA that's found in the free proteins and RNA ribonucleoproteins here. So this is Mark's work in the lab. If you do small RNA sequencing, first we will look at the host RNAs. We started with green with the microRNAs, but they're just one small flavor of the RNAs that are circulating on LDL. For host RNAs, the majority come from ribosomal RNA fragments. They're in purple. But what's really striking with the sequencing data is that we realize that a majority of the small RNAs on LDL are non-human. So only about 5% of the RNA on LDL is from the host. There's probably some reservoirs of RNA that we can't sample at this time due to terminal chemistry of the nucleotides on the RNAs in our library prep, or potentially methylation or RNA modifications that prevent the RNA from being sampled in our sequencing. But the majority of the RNA is non-host. So what kind of non-host RNA is it? Well, if you align the RNA to a ribosomal RNA database, you see that a large bit of the RNA comes from bacteria. If you start to align the RNA to different species, the genomes of different species, you realize that the RNA is coming from bacteria, both in our microbiome as well as our environment and potentially diet. There are other types of RNAs there that come from fungi and other microbial sources. If you look at the types of bacterial RNA there, the largest phylum is from Proteobacteria, here in purple. And this is very consistent. If you were to isolate lipoproteins from either humans or mice, or ApoB lipoproteins or HDL, almost 100% of the time, the dominant phylum is small RNAs from Proteobacteria, with Actinobacteria and Bacteria DTs and Firmicutes having less of a contribution. This is interesting because in the human microbiome, Proteobacteria is usually excluded. In health, Proteobacteria have minimal colonization within the microbiome. There's a few sites in the body where their Proteobacteria can grow. Now in states of IBD and other diseases, Proteobacteria will flourish. But what we think overall is that the lipoproteins are transporting products of dead bacteria. So there's not really a correlation between the microbiome and bacteria that are actually in the gut and what's circulating on the lipoproteins. We think that what are on the lipoproteins are actually, like I said, the products of dead bacteria that have been excluded. Okay, so early on, we realized that we needed to prove that the RNA on LDL is the inflammatory cargo, that we needed to remove the RNA from the LDL and experiment. And so we pulled out an old method from about 25 years ago from the Brown and Goldstein lab, and we started making reconstituted LDL. So a lot of people on the call here have made reconstituted HDL. Reconstituted LDL has its own set of challenges, but during the process of reconstituting LDL, you remove the RNA. So here's a bioanalyzer tracing of native LDL with the small RNAs. For the most part, the RNAs are between less than 50 nucleotides in length. And then if you do the reconstitution process, it removes those. You can use PCR to kind of validate that candidate RNAs are no longer on your reconstituted LDL particles. And then using our cyto assay, we can kind of demonstrate that, again, that the reconstitution process removes the small RNAs. These lipoproteins, these reconstituted LDL, they perform just like native LDL. They're the same size. We reload them with a cholesterol ester to maintain the core. That we use a fluorophore on the cholesterol ester so we can monitor LDL uptake. So the reconstituted LDL are taken up by macrophages similar to the native form. But what's interesting is that reconstituted LDL lacking the small RNAs fail to activate the macrophages and induce cytokine secretion. So here on the left, we have the mRNA analysis that native LDL turns on IL-1 beta around 5-fold, IL-6 around 4-fold, and a little bit of TNF alpha. Then at the protein level, after 24 hours in the media, using ELISA's IL-6 is significantly increased, as well as TNF alpha. However, in blue, the reconstituted LDL that lacks small RNA fails to induce the cytokines. So this is really exciting data for us. And where we're at right now with this is now we're reloading the reconstituted LDL with different forms and of RNA and different RNAs that are labeled so we can trace the RNA, but also rescue the stimulatory effect of the LDL. Because LDL has both human and non-human RNA, we're beginning to ask the question, well, are all RNA inflammatory? Is it something special about the non-host RNA? And so we'd isolate RNA from E. coli, and we would use a special protocol to enrich just for the small RNA. And when we take THP1 cells and we'd hit them with either THP1 RNA, so self RNA, human RNA, or E. coli RNA, there's only the E. coli small RNAs that would turn on the inflammatory gene expression. Now, one caveat to this is there's potentially some endotoxin or maybe something coming from the E. coli and the RNA isolation that would turn on the cytokines at the mRNA level here that we're trying to address. But overall, we think that there's something special about the non-host RNA that's stimulatory. However, extracellular microRNAs and exosomes have been demonstrated to be pro-inflammatory on TLR7, so it's something to consider. But we think that there's something unique about the non-host RNA. So here we are with kind of a break in the talk, a little bit about the mechanism. We think that the LDL microbial RNA is taken up by the macrophages. The model is it activates Telopreceptors, the endosomal Telopreceptors, and drives inflammation, and that releases cytokines and promotes the growth. So how we got to Telopreceptor 8? So Telopreceptor 7 and 8 are the endosomal single-stranded RNA sensors. They're part of the nucleic acid sensing family of Telopreceptors, which includes TLR3, TLR13, TLR9, which recognize other forms of nucleic acids. There's usually two kind of forms of Telopreceptors. There's the ones on the plasma membrane that obviously recognize a lot of the pattern recognition receptors on the plasma membrane, but also nucleic acid sensing ones on the endosome. All right, so how we got there is we would treat the THP1 cells with bifidobacterium A to determine if we're dealing with a surface or intracellular signaling process. Bifidobacterium A is a proton pump inhibitor that disrupts the endolysosomes. Hitting the cells with LPS to activate the surface TLRs was not affected by disrupting the proton pump. However, hitting the cells with LDL or the single-stranded RNA positive control here failed to turn on the cytokines. And so this suggests to us that we are dealing with the intracellular endolysosomal process. We used a calcium chelator as well that disrupts total TLR signaling, as well as many other processes in the cell. But this kind of, again, supported the intracellular Telopreceptor more so than a surface plasma membrane receptor. We then cloned each of the Telopreceptors and overexpressed them one by one in HEK cells using the NF-κB luciferase reporter. We would also co-express the UNC93B1, that's a typo there. And then we'd hit the cells with either the cognate ligands for each of these Telopreceptors or hit them with LDL. And very quickly we realized that only TLR8 was sensitive to LDL activation. And this is for native LDL. So TLR2 can be activated by oxidized lipids here, but this is for the native LDL activation. Surprisingly, TLR7 was not activated as much as TLR8. So this is one of the early clues that we were dealing with something related more so TLR8 than TLR7, which are very closely related. All right, so to demonstrate that TLR8 is the receptor for LDL activation, we used siRNAs. So we would nucleophect our Th3-1 cells with either scrambled siRNAs or siRNAs and knock down TLR8. Our knockdown was quite effective with this transfection protocol, and it worked. So LDL would turn on IL-1 beta in the presence of either the shock or the scrambled control, but then there was a significant block in LDL stimulation when TLR8 was suppressed or knocked down. And this is for all three, IL-1 beta, IL-6, and TNF-alpha. At the protein level, we observed the same thing. So IL-6 and TNF-alpha in the media were also significantly, or the induction of LDL was blocked by knocking out TLR8. So there's also very specific TLR8 small molecule inhibitors. So one is CuCpt9A. So we encapsulated this drug, a small molecule, into nanoparticles and delivered it to THP1 macrophages. And the small molecule worked very much like the siRNAs, where the native LDL would turn on the cytokines, but the small molecule inhibitor in the nanoparticles would block this effect. This was observed both at the mRNA by PCR and then at the protein level by ELISA, where the native LDL induction of these cytokines was completely blocked by the specific TLR8 inhibitor delivered by the nanoparticles. All right, so this is a little fun part of the story. We have been studying micronase for a while, and we've been using LNAs to inhibit micronase in a targeted manner, so a very specific kind of an antagomer approach. And we realized that just the chemical conformation of locking those nucleic acids, so the locking the nucleic acid here at this position provides increased stability. There is generally resistant from RNase degradation. It also provides, as far as targeting micronase, it provides a little bit more efficacy. But we realized that this lock conformation, the chemical conformation here, actually serves as an antagonist to TLR8. And so we use just the scrambled control A sequence. It's called a non-targeting because it's a scrambled. It doesn't have a known antisense, micronate target in the cell or anything like that. So we call it non-targeting, but it's just a QIAGEN or exicon A scrambled sequence. But this conformation blocks TLR8 activity. And so that we use it the same way we use the siRNAs and the small molecule inhibitors. If you pre-treat macrophages with this LNA, it completely blocks LDL's induction of the cytokines at the mRNA level as well as at the protein level. So here's some Western blots showing that the LDL was taken up by the macrophages, where you could start to see ApoB protein within the macrophages. You get stabilization of the PLIN2 protein. You get an induction of IL-1 beta, which is blocked by the LNA. And then in the media, you do see a complete inhibition of LDL-induced cytokine secretion after 24 hours by ELISA. So these are 3 different loss of function studies on TLR8 that we felt really demonstrated that TLR8 is the receptor for LDL activation of the macrophages, or native LDL activation of the macrophages. So I think one of the things that's held back TLR8 research over the last decade is that early on, initially people were claiming that TLR8 was non-functional in mice. I think over the years, that argument has waned a little bit. But we took macrophages out of the TLR7 knockout mice, and LDL, native LDL, was able to induce IL-1 beta and IL-6 in those macrophages. However, those macrophages are not sensitive to the sequence that we're using, the single-stranded RNA sequence that we're using as a positive control. So potentially there's other small RNA sequences that might be present on LDL that are driving this in the mouse. We went back to our NF-kappa B luciferase reporter to circle back and to compare human and mouse TLR8. So we overexpressed human or mouse 7 and TLR8 in these cells and hit them with different ligands. And human TLR7 is not activated by LDL. It is turned on by some of these nucleoside analogs. Single-stranded RNA 40 is our positive control for all of the So mouse TLR7 is not activated by LDL, but mouse TLR8 is. And so we think that mouse TLR8 is indeed functional and activated by LDL. So we decided to go look in vivo to target TLR8 in vivo as far as a model of atherosclerosis, reducing atherosclerosis. So we wanted to use these non-targeting lot nucleic acids to inhibit TLR8's detection of LDL small RNA. So first we needed to demonstrate that these bacterial RNAs are present in lesion. That was a big step for us. So we generated atherosclerosis in ApoE mice, high-fat diet over, I believe this was a short time point of 4 weeks. We isolated cells out of the lesion through FACS, and then we isolated RNA and did PCR or did sequencing and confirmed by PCR. And what we found robust detection of microbial RNA within macrophages and within the lesion. So these are 13 different pools of mice here where these are CD11b positive cells that we sorted out of the lesion, and they're highly enriched for bacterial small RNAs. This was sequencing where we couldn't really tell if the RNA was coming from the environment or from the microbiome, because a lot of these fragments come from ribosomal RNAs that have high conservation between species of bacteria that are present in both the environment and the microbiome. So we don't know exactly which is a donor bacterial species, but overall, the total level is quite high. This is 150,000 reads per million within the macrophages. And then we identified some candidates, and we did PCR for a handful of candidates showing that compared to just water control, that there's quite a bit of detection of bacterial RNAs and from the cellular RNAs that we isolated. So this is our in vivo kind of strategy model, is that we're going to use LNAs to just inject in the mice and block this mechanism. So we first did an early athero study. We put ApoE mice on a high-fat diet for 4 weeks. We injected weekly. And surprisingly, after 4 weeks, we saw about a 30% reduction in Olredo with this. So we saw almost rapid mobilization of the neutral lipid from the lesions, which has been very consistent for us. What's interesting is that this treatment of the LNA at 30 mg per kg did not affect any plasma lipids, so there's no change in total cholesterol or triglycerides in these animals. There's also really no change in macrophage content. So this is F480 stain, and there's really not a huge difference in the macrophage number here, which we'll come back to in a second. looking at some of the collagen and fibrosis, we really didn't see a big change there. Really, our big phenotype that we're observing is just a mobilization of the neutral lipid, the olradosane. So we then kind of switched to a regression model where we LDL-R mice on a Western diet for 14 weeks, and then we would convert them to the chow diet and inject weekly with either saline or LNA, and then look and see. And we had both male and female mice, and we found that the LNA blocked the additional growth of the lesions. It did not reduce the lesion content compared to baseline when we did the diet switch, but the saline mice continued to develop athero, where the LNA mice did not, as far as olradose staining in lesion area. But again, we failed to see changes in macrophage content, even though the olradose was changing this model. So we started doing single cell sequencing, and this has really been great for us to use this tool to ask the question about some of the macrophage phenotypes with this treatment. So this is our cell counts that we're able to get different pools of mice with our single cell sequencing. This is with we did FACS sorting of the CD45 positive, CD3 negative cells, and we used a 10x system for this. Overall, if you look at the gene expression in cells that were called to be macrophages, we found a significant reduction in pro-inflammatory genes of just kind of pan macrophages that were called. So IL-1 beta, NLP3, CCL3, some of these other inflammatory genes were significantly downregulated. We then did a reclustering that really dive into the macrophage subphenotypes, and we found, I believe 14 different macrophage subphenotypes that we were evaluating. And kind of going back to the recent meta-analysis paper and the half dozen single cell sequencing papers over the last 3 years on athero, we really kind of use all that information that help us, and we kind of went one by one with each of these clusters and tried to kind of name them with by hand. And what we found with the LNA treatments is that overall the number of macrophages didn't change, but the polarization did. So the LNA treatments decreased the amount of inflammatory and TREM2 foamy macrophage phenotypes that we found. But it increased the number of macrophages that we would call proliferating or cavity macrophages or repair macrophage phenotypes. Those were all increased with the LNA treatments over here. So we think that the macrophage number doesn't change, but obviously the inflammatory macrophages we think are going down, and the proliferating and cavity macrophages are compensating. So kind of in summary, we think that microbial small RNAs on LDL activate Telopreceptor 8 signaling, and this drives pro-inflammatory gene expression in macrophages. We think it reorganizes the macrophage phenotypes, which ultimately promotes healthy removal and efflux of cholesterol from the lesion. We don't see a consequence of plasma cholesterol increasing, and to date, we don't see a lot of evidence in other tissues of where the cholesterol is going, but we are actively looking for that. But we think that these microbial RNAs on LDL are a pro-inflammatory stimuli, and we think that they can be targeted. And so we are really going after TLR8 now. We think that TLR8 has been ignored in the athero world, probably because people thought it was non-functional in mice for a long time, but we think it just has slightly different function than what's reported. So this is a review last year kind of in Trends of Cardiovascular Medicine, and these are all different pathways that likely influence or contribute to athero-associated inflammation. And we don't know exactly how the LDL microbial RNA fits into each of these, but you can come up with a pretty plausible role for LDL small RNAs in each of these little tracks I've highlighted here, whether it confers some of the environmental contribution to this. There's clearly a relationship with HDL. I didn't talk about it today, but we think that HDL will remove the RNA from the lesion in what we're calling a reverse small RNA transport pathway. There's also, obviously, there's a relationship with the gut microbiome. The lung microbiome might be more important than the gut. That's to be determined. And then obviously chronic infections and inflammatory states, as well as things that could affect the gut barrier function, whether it's HIV or inflammatory bowel disease, things like that. So, we think the most important question right now in the field of athero is trying to identify what is the kind of upstream stimuli for inflammation. And, we think the barrier to this is really the discovery of new stimuli outside of cholesterol and bioactive lipids. And we think that this falls into that category. And obviously we think this overcomes some of those barriers and really just kind of fits in with this kind of overall shifting paradigm of athero that has kind of come about over the last decade. So this is a picture of our kind of group. This is Ryan's project I just described today. Mark Hasselberry has contributed to this. Danny is our lab manager. She's a fantastic lab manager. Some of the samples we got from Japan. Maki is a good collaborator of ours. And then I just want to highlight Mac Linton. He's a very close collaborator in our atherosclerosis research unit. This project has a lot of computational analysis, and Tiger in the Center of Quantitative Sciences at Vanderbilt is in Marisol are both integral part of our research program. So with that, I'm happy to take any questions, and feel free to email me if you think of something after the talk. So, thank you.
That was a wonderful introduction to the topic for us, Casey. Thank you so much. So we're going to take some questions. And as you know, yes, Katie is leading the way. First question from Katie. And then you can just raise your hands, please.
Yeah, I can't raise my hand because of, that was great, Casey. Thank you. I have a few questions, and actually right at the end, it kind of sparked one as you were summarizing sort of all of the things that are known about, the contributors to atherosclerosis, and you mentioned environment and microbiome. And so what's the source of your LDL in terms of, does it come from different places, different donors? I mean, I know at some point you switched to, I don't know when, during the presentation you were using recombinant recombinant sort of LDL and things like that. Have you noticed, do mice in different, we all know that different mice in different environments obviously have, lots of different things that are different, but have you looked there in terms of different microenvironments, different, microbiomes, that kind of thing?
Well, we have. So the lipoprotein profiles are sensitive to their environment. So we have different mouse rooms at Vanderbilt, and those different rooms have different profiles. We can order kind of wild-type mice from multiple vendors. They arrive here with different profiles, and then when we kind of let them hang out in the same room for about 30 days, they will converge. Those profiles will converge.
Right.
So that makes a lot of sense. Other things don't make as much sense when kind of messing with different environmental factors. So we don't really know exactly what's going on with the environmental contribution to that, but there is one. Mice and humans, the profiles are similar overall. Different bacteria, if you dial into the species of bacteria that are contributing, can be different. But I think overall, at the phylum level, they're very similar. We have, over the last few years, tried to collect samples from different collaborators across the world, different environments, just sending in plasma that we would do density gradient. And we are seeing some location-specific differences. We see differences in RNA from Japan versus Nashville. We don't know how that would factor into health, like, your environmental impact on your cardiovascular health yet, but there are RNA differences.
Cool.
And yeah. Eastern Europe has maybe not Eastern Europe, but samples from Austria have a very unique bacterial profile that's different from Nashville.
Okay.
Yeah.
Cool.
I think Adil was the next one. Yes, Adil.
Hi, thanks. That was a really great presentation. Really interesting. So I'm wondering if you know or have thought about what's special about LDL versus HDL or VLDL. Is it packaging with the specific apolipoproteins, or is it just because it's potentially more cholesterol rich than some of the other lipoprotein particles?
Yeah. So we found out about a year ago that it's a protein. It's not the lipids that binds the RNA. So I thought for a long time it was phosphatidylcholine, but that didn't pan out. So HDL, we think ApoA1 is an RNA binding protein. It's not the lipids there. And on LDL, we think it's probably some kind of protein that binds it. We are obviously looking at ApoB. So yes, LDL and HDL both have RNAs, but they might have different RNA binding proteins. I know that some people are studying chylomicron RNA. We don't find RNA on chylomicrons, but other people I know have grants on that and things. So I guess to be determined.
Yeah, the RNA binding proteins is probably key to this. I agree.
Yeah.
Okay, so Lale.
Hey, thanks. Casey, it was a great talk. Thank you. I have a quick question. So this small RNA on LDL activating macrophages, macrophage inflammation is interesting. Do you think this is specific for macrophages, or other immune cell types also respond the same way, like dendritic cells or T cells?
Yeah, we do. So any cell that could bring in and catabolize LDL, particularly the cells that have kind of non-saturable pinocytosis, that phagosomes that could form, or I guess the phagocytes that would take up this. The cells that have TLR8 would be the ones that would probably have the most stimulation. So myeloid dendritic cells, maybe some cells that were not, maybe podocytes in the kidney. That there's some kind of random cells that have TLR8 that we don't necessarily think have taken up LDL. So it's kind of it requires both. It requires LDL phagocytosis, and it requires the TLR8 expression in the endosome. Now TLR7 might do it in maybe the plasmacytoid dendritic cells, but and monocytes is not as strong. The monocytes have a little bit of TLR8, but then it's like when they differentiate in the macrophages, TLR8 really turns on. I think that's when the system becomes hot. Monocytes maybe can do a little bit of this, but it needs that TLR8.
Okay, thank you.
Claudia?
Hi, Casey. It was a tremendous talk. I'm really excited about this new link between inflammation and lipoproteins. And you're absolutely right. TLR8 is always forgotten. And I think it was really, really spot on. I cannot praise this more enough. But I have a question, is more like curiosity. So do you see the TLR8 expression? You partially answered now to Lale, but I wanted to know specifically on the macrophages, if you find it expressed in the inflammatory or on the regression emerging phenotypes, and how do you see this temporarily? Is that an activation in tissue? And as an add-on, I would say, are you sort of tested other bacterial components that can be within the lipoproteins, and maybe that can have other contribution to, maybe not to this signaling, but maybe a signaling recognized by other more well-known TLRs like 2, 4, because I think that would be very exciting to have the LDLR as carriers. So,
yeah. So we actually haven't studied, TLR8 expression the regulation of TLR8 in some of the macrophage polarization. We kind of just make sure it's there, and we are starting to think about there's some SNPs in TLR8's promoter that are interesting. It's related to metabolism and cardiovascular disease. So we're just kind of learning about how TLR8 is regulated, both at the transcription and promoter, but also the degradation, the turnover. Like, how is this system turned off? I think is going to be a big role. So we really don't know a lot about TLR8 as far as the different subphenotypes, but as far as the other proteins and bacterial cargo that could come along on LDL, that is certainly in play. We use the HEK cells to really isolate into specific TLRs, but then going back to the macrophage that has most of them, we kind of used the specific inhibitors against TLR8 and the sRNAs against TLR8 to really drill down on TLR8. We didn't really see a lot of evidence that other TLRs were in play with our native LDL, but I think maybe we used LDL from disease models or oxidized that those observations would be obviously different. But yeah, we think that LDL could take up other bacterial products, in LPS, or maybe some bacterial proteins that could come along that could stimulate the macrophage as well. So we're not ruling out, we're not saying that RNA is the sole stimulation. That's my dog, but yeah, a lot to learn there about TLR8.
Thank you.
And thank you, Mabruka. Yes.
Thank you very much. That was really very magnificent talk. So apology in advance. I'm afraid I missed the first part of your talk. So where the activation for TLR8 is happening? Is it happening in lesion atherosclerosis, or do macrophages get activated when they've been in circulation or in myeloid, in their myeloid progenitor stage before they get into differentiated?
Yeah. So Andrew Murphy and his group had a really great study about 5 years ago now, where they would look at basically monocyte differentiation and polarization to different phenotypes, and they found that the native LDL treatment of monocytes really pushed those macrophages to different phenotypes, or push those monocytes to different phenotypes. So yes, there's probably this story probably starts in circulation to some degree, but I think it really is turned on when macrophages are sampling their environment in the lesion, and LDL is just accumulating to such high degree, and that you have this penocytosis. And I think that's where the RNA can start to accumulate to to a high enough degree to drive the signaling.
Have you looked for IL-1 alpha or IL-1 receptor antagonist?
Yeah, that's a great question. We haven't. As far as I know, they might have done some PCR on it, but things like that. But yeah.
One last question: is there, there is a huge clinical evidence between pneumonia, especially hospital-acquired pneumonia, and the incidence of myocardial infarction. So, have you looked for plaque erosion or plaque rupture with TLR8 activation?
No, we haven't. Peter Livy just gave a talk at Vanderbilt yesterday. So this erosion thing is really exciting, and I don't know, it's kind of somewhat new to me. So I think we start talking about like neutrophils and nets and capturing these things. I bet it's capturing a lot of this extracellular RNA, and so if there's a single-stranded RNA extracellular, stimulus, is probably going to get caught up in those nets. And so we don't know about the relationship to erosion just yet, but I bet there's something with this there.
Interesting. Thank you.
I guess I'm the last one to ask questions, Casey. So once again, thank you for this great talk. I really wanted to learn about this subject, so thank you. And my question is, since RNA binding proteins might be really important for this, have you sampled the RNA binding proteins in these endosomes at the vicinity of TLR8 in your stimulations?
That's a great question. We have not, but there's tools to do that. This cross-link tools and with sequencing and mass spec and things like that. You could get at that question, and we'd love to do that. And yeah, it is an exciting toolbox with a lot of tools to answer these questions.
Yeah, so it's definitely doable. Another thing that occurred to me is I remember that in axons, endosomes are sites where local translation happens. So do you think the purpose of having TLR8 and these microbial small RNAs might also be relevant to local translation, important for endosomal trafficking, for cholesterol metabolism? So local translation changes might be important to communicate with the microbes and macrophages and other immune cells.
I don't know a lot about that topic. That's really interesting. This essentially, this would still be kind of on the outside of the cell, within the endosomal, core, if you will, I guess. So I don't know a lot about endosomal translation. If TLR8 kind of maybe during its as it's moving from the ER, to during its own maturation and localization to the endolysosome, there's a lot there about UNC 93, 1B and some other regulators of getting TLR8 to the endosome. That maybe that's in play, but I think once it gets to the endolysosome, that's where it meets the LDL cargo. And so I don't know about along that process. I don't know where the translation would occur and how the RNA would affect that. But
yeah. So a little bit from the neuroscience field, but definitely endosome can play a role in local translation, and that could affect endosome function. So maybe that's why TLR8 is there to communicate with the distant microbiome. Just a thought.
Yeah, that's great. Yeah.
All right. Well, any other questions, or should we part for the weekend? This was great. And I also wanted to say that Casey would like to do a session for the metabolism trainees. So I know Mabruka is here, Adil is here. So it would be wonderful if you guys get in touch with him. So we'll see more of you, Casey, hopefully.
Okay, good.
All right. You guys have a great weekend. Thank you for joining.
See you, Casey. Thanks again.
Thanks again. Bye.
Thank you. Bye.